light has the property that it can travel through our skin and bone and allow us to be able to see into tissues. And in neuroimaging, we take advantage of this fact in order to be able to see into the brain and understand how it works. One of the things that we're interested in looking at is the levels of oxygen in blood. So as some of you might know, when oxygen binds to hemoglobin in blood cells, the color of the blood is red, and is otherwise when there's less oxygen binding to hemoglobin, the blood is blue. And I'm going to show you a small video that demonstrates this. So as oxygen gets used up by cells around our veins and arteries or wherever it's being delivered, the blood changes color. And one of the properties of near-infrared light is that it allows us to see this and to provide us with measures of changes in blood oxygen levels. And so near-infrared spectroscopy is a technique which actually takes advantage of these principles. And the way it works is that you can actually place sensors on a subject's head, and where near-infrared light enters the head at one point by means of a sensor, which is then absorbed by the skull and the brain tissues, and whatever is not absorbed is returned back, which is then detected at this sensor placed a short distance away. This can basically provide us with information on changes in oxygen levels in those parts of the brain, and therefore provide us with a measure of brain activity. And this is in essence what near-infrared spectroscopy is. And to date, it's been used for a number of different areas in the field of neurodevelopmental research. It's been used to investigate typical brain development in areas such as infant social cognition, to understand language processing and object processing, but also in atypical brain development in disorders such as autism spectrum disorders and in malnourishment. And previous work from the BASIS study that takes place at the Birkbeck Babylab, where infants at high risk for autism are studied, which are infants that have an elder sibling that has been diagnosed with autism. Results from that study have shown that NIRS has the value of providing us with interesting information in that might be happening in atypical brain development. So I'm going to be referring to some groups in the next slide, which I'll just define here. So there is the low risk group, which are infants that have no familial history of ASD. The high risk group, which does not receive an ASD diagnosis at 3 years of age, and the high risk group, which does receive an ASD diagnosis. So the results from this study have actually shown that in infants that receive the ASD diagnosis, the high-risk infants, which is this purple line you see here, the brain responses to the social condition are actually quite different in the two groups compared to that high-risk group that does receive an ASD diagnosis. And it's important to note that there's no difference in behavior between these groups at the 4 to 6 month time point, which is when this data was collected. And what this is showing us is that basically NIRS can potentially provide us with important early markers of atypical brain development. So typically, two or three wavelengths of light are used to measure changes in oxygen levels. And while this is really useful, it doesn't give us a full picture of what's happening in the brain during activation. So my colleagues at UCL have actually developed a new type of near-infrared spectroscopy, which uses a wider spectrum of light, which can provide us with information about changes in oxygen levels, but also other things such as energy usage in the brain, which is otherwise known as metabolism. So I'm just going to play a short video for you where we zoom into a neuron to see how a cell uses glucose and oxygen to produce energy. So we're zooming into an organelle known as the mitochondrion, which basically uses glucose and oxygen that's present in the environment to produce energy, which the brain cells then use. So it would be really useful to be able to measure while typical NIRS would give you measures of the oxygen that's being delivered, the broadband NIRS would allow us to capture this whole process as a whole. And so, just to give you a background of what the process is, so during metabolism, there are a number of molecules that are involved in energy generation. And I've got another little video for you to show you where this is. So there are a number of processes involved, and one of them is this highly complicated looking process. But what we're interested in is this enzyme. Sorry, my mouse is lost. This enzyme here called cytochrome C oxidase, which actually absorbs near infrared light to our advantage, and it changes color based on how much energy is being made and used. And so using broadband NIRS, we can actually have an idea about how energy is being generated and used. So I'm not going to bore you with all this research, but there has been a lot of research that's been done in piglets, in neonates, and in adults, which actually shows that cytochrome measurements, cytochrome is the enzyme in the mitochondrion that I was just talking about, they actually correlate with markers of cellular function. So what this is telling us is that it's actually a more direct marker of brain activity, and it may be particularly interesting to measure in conditions where neurological pathways might be impaired. So some of you might remember that I did the NEST infant study almost 2 years ago now, where basically we wanted to use this new system that was developed at UCL to see whether we could obtain these measures of changes in oxygen level and metabolism in the 4 to 7 month old infant age range in response to functional activation, which is showing them videos of women singing nursery rhymes and that kind of thing. So this is just a picture of an infant wearing the custom-built headgear, where you can see the fibers are positioned on the head, and it was positioned over the right temporoparietal region. And we studied 42 infants, and we used 2 different conditions. So one was a social experimental condition, which consisted of videos of women singing nursery rhymes, such as Incy Wincy Spider and Wheels on the Bus, and the non-social experimental condition, which consisted of videos of mechanical toys, that kind of thing. And I'll just show you some of the results. So here is a picture of all the fibers on the head. And we found that in response to the social condition, the biggest activation that occurred was over this region, which is known as the temporoparietal region, in accordance with previous studies, which is known to activate in infants in response to faces. And there's a bigger response to the social condition in comparison to the non-social condition. So there was an increase, there was a bigger response for the social condition for both the change in oxygen levels as well as metabolism. And now I'm just going to show you a video that we have created. So basically on the left, you'll see changes in the oxygenated blood being delivered to different, to the regions, those regions of the brain. In the middle, you have deoxygenated blood. And on the right, you have the change in metabolics or metabolism. And so again, just as a reminder, oxygenated blood is the blood, the blood that's being delivered that contains oxygen, so that those regions of the brain, the cells in those regions of the brain can use the oxygen to generate energy. So we expect that this oxygenated blood should be closely linked to the metabolism. So I'll just play this video for you. So this is in response to the social condition. Initially, in infants, you see an initial dip where there's a decrease in oxygenated blood and metabolism, followed by a large increase in oxygenated blood and metabolism. So there's an increase as you would expect in similar brain regions. So that was the social condition, and then this is the non-social condition. And as expected, there's not as much of a strong response to the non-social condition in comparison to the social. So in conclusion, we used broadband NIRS to measure changes in oxygen levels and metabolism in 4 to 7 month old infants. And we found a significant difference between the social and non-social conditions. And this was a very exciting study because this was actually the first study that used broadband NIRS in this age of infants in response to showing infants faces and cars and helicopters, and no previous studies have actually measured the relationship between the changes in oxygen level and energy metabolism during development. And this is actually particularly important if we are to understand the biological mechanisms that might be disrupted during atypical brain development. So while in the videos that I showed you, while the oxygenated hemoglobin and the metabolism look quite similar in the typically developing infants, we expect that maybe in the atypical brain development or in disorders, there would be where there is evidence that this relationship might be disrupted. This would be particularly useful for us as a technique. Yeah, so special thanks to all the parents and infants who participated in this study and everyone who helped me do this work. Thanks.
Thanks, Maheen. That was really interesting. Does anyone have any questions? You don't have to be really sciencey, like any question is.
So I tried to keep it as brief as simple as I could, which is quite difficult because it's technical.
Yeah, exactly. So I think some of you who've joined us had your babies who are now toddlers took part in this study. So do you have any questions about it? About basically your children were pioneers.
I think someone's...
It's not a question directly, but I really love to be able to read more. Is it possible to share anything you've published so that I can read a bit more?
So we are in the process of publishing this study. We haven't published yet. I'm hoping in the next month or so. And I think there's a Babylab newsletter that goes out that will have am I right, Tamsin? That will have the information about the paper, yeah. So,
yeah, should do. It's in the process of being put together. So I'm hoping that will get sent out within the next, well, I've been saying this for a while, but in the near future.
Is there a central point where you make papers available that have been generated by work at the lab? Or are they, obviously they're individually published depending on their purpose, but is there a signposting website where I could see more papers from the work of the lab?
Generally, you would find, I wish we did have something like a central reservoir of all the research that comes from the lab, but I think because there's so much, it tends to be on the individual researcher's page. So on the CBCD website, if you go to the people pages, and if you click on Maheen or Brittany or Rihanna or whoever, their papers they should be, I think this is right, guys, mine are, but their papers should be listed. So their most recent ones will be on top. And then it should be a direct link to the paper that you can access, hopefully. Is how it works. And then if anything, sometimes when our research is highly topical and if it gets picked up in the news, then that would be in our news section of the website. So again, our website is probably the best place to go. you're very serious. Sorry, I'm being distracted by your gorgeous baby. Yeah, so the website is the best place to go.
Thank you. And thank you, Maheen. Sorry to hijack your questions, but that was a fantastic presentation and really clear and super. It's just really amazing to know about your work and everybody else's work as well. But anyway, so sorry for hijacking your questions slightly. I'm sure the people have got more pertinent things to say.
No, that's great. Thank you for your interest.
Sorry, just to add on where to find papers, I don't know if you already follow the social media, like the CBCD social media accounts, but I think often they post if there's a new paper out. So that might just be another good place to kind of see what's coming out all the time, which you follow already.
Did you say?
Yeah, Brittany, you control those pages. You maybe have, is it more Facebook or Twitter, which is better to follow?
We post them on both, so we do Twitter and Facebook. And yeah, if a paper came out, we would do both. Mm-hmm.
So we've just had, Emily Jones has just had a paper published in the Nature journal, which was tweeted and posted on Facebook and Twitter, and is also available on our website. Any more questions? Well, Maheen, thank you so much for sharing that with us. It really was interesting, and I particularly liked all the video demos that you had. That really sort of brings the hard science to life, so to speak.
Yeah, so I have to give credit to my colleagues at UCL who actually ran this project called Metabolite, which was funded by the Wellcome Trust, just a project specifically for public engagement. So they came up with these brilliant videos, because otherwise it's so difficult to show these things simply. So I can't take credit, but it's really good what they've done.
Yeah, it's amazing. But thank you for sharing that with us as well.
Yeah, thanks for having me.
A pleasure. So we obviously have our lab research, but we do also have some online studies. We've got 5 going now, and we've got more that will be joining over the next couple of weeks. Just to quickly highlight them, we have got ones basically for all ages, but for parents that are joining us, Anna, one of our, actually she's finished her PhD, but she has put together a survey for parents. It's a really short survey just to get your feedback, your opinions on how you feel about data sharing. So when you take part in research, so both your personal data, but also your child's data. So if you have time to fill in that survey, that would be a huge help to us. We also, I'll go down in order age-wise. So then we have Brittany, who is still looking for 11 to 18-year-olds for her study, which is it just takes 30 minutes, and basically it's looking at how noise can impact learning. So if you have a teenager or a preteen, please consider asking them to do that, because my kids did it. It's really fun. They basically play some brain games while listening to noise or not noise in some headphones. And again, this is all done from the comfort of your home, so it's very safe. Then we have Claire. I'm not sure if she's with us today, but she is really interested in how cartoons can impact attention and learning. So she is looking for basically 12-month-olds, so 1 to 6-year-olds. She has a really short 10-minute game that seems like a memory game, and all the little kids I know who've done it have liked it so much they've asked to do it again. So if you have someone of the right age, please get them to play that game. And then we have Ellie, who has the app that she's developing for proper infants, so for 0 to 12-month-olds, and that's just sort of a way for parents to help identify, I guess, standardized developmental milestones. And again, it's all done from home. Yeah, and like I said, there's a couple more that will be joining us shortly, so please check our website, our online studies page to learn more all about that. So that's enough from me. To finish off, Maheen has found this fabulous song, which because I don't have an infant anymore, it's not that new, so those of you with babies might already know this, but it's called The Happy Song, and I will share that now. So let me just share my screen. Here we are.
I know this by heart now.
Yeah, I bet you do.
Yeah.
Can everyone see that?
Yeah.
Okay, great. And let me make it big. Here we go.
submarine. Choo-choo train, aeroplane, oh, we gonna fly. I just a da-da-da, in every day, wherever we are. So high in the sky, and deep in the ocean, through valleys and hills, away we go. So up in the sky, and deep in the ocean, through valleys and hills, away we go. You little monkey, staying up late, who purrs like a cat when they get their own way, who then turns into a lion who likes out there. In the sky, and deep in the ocean, through valleys and hills, away we go. In the sky, and deep in the ocean, through valleys and hills, away we go.
All right. That's it from us now. So thanks everyone for joining us. We have got 2 more talks lined up, so we'll be back for the next 2 weeks, and then we'll have a break for the summer. So thanks again for joining, and hope to see you again next week. Take care. Enjoy the sun.
Bye.
Bye.
Bye.